So my name's Goodeve. I'm from Sheffield in the UK, and I'm going to talk about pathogenic or not, classification of genetic variants in haemostasis, and the same principles can be applied to other disorders. So these are my disclosures. So genetic analysis is commonly used for Hemophilia A and B to determine the variant responsible for the disorder. For von Willebrand disease, we may have some reasonably good phenotypic classification to help us understand the disease type, but where that's not the case, genetic analysis can help to clarify the disease type. And we can also apply genetic analysis to a number of other hemostatic and platelet disorders that will benefit from understanding the variant responsible, or variant or variants responsible for the disorder in that individual. So why would we want to look for sequence variants? To help determine the cause of the disorder in the patient, to determine any associated consequences of that variant, for example, risk of inhibitory antibody formation and phenotypes like the discrepancy between 1 and 2 stage assays in factor VIII. Because many labs will only do one of the factor VIII activity assays, they may not realise that there's a discrepancy between those two, but we know that there's a number of variants that have been identified with that discrepancy. So through genetic analysis, we can predict that that will be present. And once we've identified the variants in an index case, this can then enable inheritance of the 1 or 2 sequence variants in a family to be determined and to look at their risk of inheriting the same disorder. So I've put on a few of the genetic analysis techniques that could be used for looking at these variants. Conformation-sensitive gel electrophoresis is one of the techniques that has been used to seek variations in a fairly cost-effective manner prior to Sanger sequencing to identify a specific variant. PCR and Sanger sequencing is still routinely used in many laboratories, but others are changing over to next-generation sequencing, which can either use polymerase chain reaction to amplify the regions of interest, or use sequence capture through hybridization to identify the regions of interest before sequencing. And as Barbara mentioned, we can use multiplex ligation-dependent probe amplification for large deletions or duplications in at least in some of our genes of interest. So this is standard process for genetic analysis. So generally we extract DNA from blood samples, examine that DNA for sequence variants using our method of choice, and then we need to determine the pathogenicity of the variants that we've identified, which can be the tricky part, and then try to report those findings to the referring clinician. So Barbara's previously mentioned the American College for Medical Genetics guideline on variant interpretation. In the UK, we use the one that was written by our society and also by the Dutch society. So it was a collaborative process between the two. And for this process, we analyse a number of different classes of evidence about the variant to help us understand whether it's likely to be pathogenic or not. So these include the extent of the analysis. So for most genes, for factor VIII and factor IX particularly, we would look at the entire coding sequence of the gene. For von Willebrand factor, if we had some good starting information from the phenotype, we might analyse a specific region of the gene. We can look at population frequency, and I'll come back to an example from the exact database in a little while. As well as looking at the sequence databases, which are always a good place to start looking for the variants. If we can't find them on those, we can undertake a web-based literature search for the variant. Some of the mutation types are very likely to be pathogenic, so frame shift and nonsense mutations. Where we've got amino acid data to look at, we can use a number of different tests to try and help us work out whether they're likely to be pathogenic, and similarly for splice site mutations. And then there are a number of functional analyses that can be done if we have a variant that somebody else has kindly previously expressed in vitro, or maybe we can undertake that work ourselves to try and look for a consequence on the protein production. If we've got a variant that looks like it might affect the messenger RNA, we can choose to investigate the messenger RNA to see whether it does have that impact. We can look at genotype-phenotype correlation and co-segregation within the families. And occasionally we have de novo variants with de novo disease in a family. So there are a number of different lines of evidence that can help to tell us whether the variant's likely pathogenic. So these are the classifications used by the Association for Clinical Genetic Science classification. So 1 is a neutral variant, 2 is probably neutral, 3 are the annoying class in the middle, where they're unclassified and there's generally not enough evidence to lead you one way or the other. 4 are probably pathogenic, and 5, there's good evidence for their pathogenicity. So this is the scheme that we use to help us classify the variants that we identify. So I've just got an example of a family here with 2 males in the family with haemophilia, where they've been sequenced and we've identified a sequence variant, which is the 6506G2A, predicted to lead to an arginine 2169 histidine mutation. So we can look on the Locusspecific databases to see possibly the variants present in several patients. In one patient only or not previously recorded wouldn't really help us. So we would hope to see it in a good number of individuals. And we can use a number of databases. So Barbara's mentioned a couple of these already. So for factor VIII, there's 3 different resources that we can query. There's the database held in London for factor VIII. There's the one in the States, the CHAMPS databases. And there's Hemobase held in Spain, which is a fairly small repository. For factor IX, we've got similar resources. So the one held in London, the CDC, and the Hemobase. And for von Willebrand factor, we have the one that we maintain in Sheffield, and there's also a Hemobase version of a von Willebrand factor database. So we have a number of resources that can be queried to see whether variants have been previously reported in other patients with the same disorder. So if we come back to our family with haemophilia A, we can look at inheritance within the family and see that we have obligate and a diagnosed carrier through the genetic analysis. This is the output from the factor VIII database for this particular variant. So it tells us it's a missense point mutation, and it's one that's been reported 103 times in other individuals with haemophilia A. And if we click on the show on here, it will illustrate the information for us. So there's sufficient information to tell us that this is really a variant that does cause haemophilia A, and it's associated with a risk of inhibitor development as well, which is useful information to pass on to the clinician. And this just shows a small number of the entries for that mutation. So we have a range of phenotypes associated with that variant, and a number of individuals, and quite a few of the individuals with this variant have a history of inhibitor. So it's one that we would warn our clinicians about, that there's a risk. Although it's generally a mild disorder for this variant, we'd warn the clinicians that there's a possibility of inhibitor development, particularly if the patient's going to be given a relatively large amount of concentrate if they're having surgery or following trauma. We can look at genotype-phenotype correlation, as we've just done, to see has the variant been seen in other patients with the same phenotype, and where that correlation is present, as we've just seen, it strongly suggests pathogenicity, especially when we get a good number of individuals with the same change. If the variant's not present on the locus-specific databases, we can try a literature exploration, either using PubMed or just searching using Google, using the gene name, the cDNA number, and the protein variant, if there is one. And that may bring up references that have been missed by the databases. And I know for von Willebrand factor, we're a little behind because there have been quite a few publications with a lot of new variants in them. So although the database curators do try to keep up, not everything will have been recorded on the sequence databases. So I've put into here a variant that is actually once on the Hemophilia B database, but it has no useful information connected to it. So if you found this variant, you'd look on the database and there's no clotting factor level on the database, and it's not mentioned on the sequence variant databases. So this one would be one of those that's tricky to try and classify, and we'd need to use some of the software predictions for this variant. So as well as looking at current nomenclature, where we use the Human Genome Variation Society nomenclature, there's older nomenclature from when the genes were first sequenced. And if you're looking for the variant and can't find it using current nomenclature, you could go back to the previous nomenclature, and for each of these three common genes that we look at, factor 8, factor 9 and von Willebrand factor, there are numbers that differ for legacy nomenclature. So for factor 8, the cDNA numbers stayed the same, but the protein started from -19 because many proteins used to be labelled from the start of any leader sequence rather than from the... They used to be numbered from the start of the mature protein, whereas they all have leader sequences. So we have negative numbers for a couple of these genes. So for factor 9, there was a numbers down to -46 for the amino acids before the mature protein started. And von Willebrand factor in old nomenclature started from the beginning of the mature protein, which is amino acid 764. So if you're trying desperately to find any previously reports of the variant, you can try using these numberings. There are a number of databases that list the variants. So we've mentioned EXAC already, but there's also the Exome Variant Server in Washington, and the NCBI lists a number of variants as well. So if the variant that you're looking at is present in one or a small number of individuals, maybe it's disease associated because it's fairly rare. If it's present in many individuals, it's much less likely to be disease associated. So I hope this shows up all right. This is a little extraction from factor 8 showing... I've put part of exon 14 on here, but just because I wanted to show the difference in the number of variants for some of these changes. So we have here a number of individuals with a missense change in them, and you can see there's only a very small number of individuals with each of those out of the 87,000 or so individuals that have been analysed. So only between 1 and 4 individuals with each of those. So they're rare variants. But in contrast, this one that affects ASP 1260, you can see it's much more common, and this is one of the polymorphic variants that are seen frequently in factor 8. So this is, for many of us, the favourite tool to look at for population frequency. This one's a little bit of a cautionary tale, where a variant that looks like it may well be pathogenic isn't. And this is all our fault in Sheffield. So Many years ago, in the 1990s, we analysed a cohort of Turkish patients, and this particular individual had type 3 von Willebrand disease, and we found a homozygous variant at 6187C to T, resulting in predicted to result in a proline to serine substitution. And at the time, there weren't many tools available for analysing amino acid substitutions. And we thought, proline to serine, yeah, that'll be pathogenic. So we suggested that it was initially, but later on undertook some in vitro expression work that actually showed it had no impact whatsoever on our in vitro expression. So the levels are the same for the variant and for the wild-type protein. It was later published as pathogenic in 11 homozygous Indian patients, but if you look on the exact database, it's present in 2.5% of individuals, so it's further evidence that it's very unlikely to be pathogenic. So take care when looking at the various lines of evidence. So amino acid pathogenicity tools can help predict possible pathogenicity, and the recommendation is to use at least 3 different algorithm types to achieve a consensus. So I'm not going to go into how any of these work, but they come under a number of different classes, and you're recommended to use tools from different classes rather than using ones that may well use a very similar algorithm. So these 3, SNPs and Go, PhD, SNP and Panther, all come under the machine learning heading. For supervised learning, we have 4 different tools, at least, within this category. These are the ones that are on our list that we use. And for sequence conservation, there's a further group. So the recommendation is to use them from each group and not ones that use very similar algorithms to each other, in the hope that you can come up with a consensus. And this just illustrates one of the outputs. This is from SNPs and Go, looking at 3 different amino acid substitutions and the predictions for pathogenicity. So for the first one, we've got a cysteine to glutamine change, where it's predicted to be very probably pathogenic, up here. The second one, similarly, even more likely to be pathogenic. And the threshold for these substitutions is for the number to be at least 0.5 for them to be classified as pathogenic. Whereas the one at the bottom, the serine to leucine, is likely to be neutral and not have any impact. So that's just an illustration of one of the tools as to how they work. Depending on the tool that you use, often they use terminology to tell you whether it's likely to be pathogenic or not. So terms such as benign, unlikely pathogenic, tolerated, possibly or probably damaging, and deleterious are used by the various different tools, and often with a number associated with it to try and tell you the extent to which they think it's likely to be pathogenic. And ideally you want those to match for a number of different tools. For in-frame deletions or insertions of an amino acid, I'm not aware of any tool to help assess these variants for now. So they're a bit more guesswork if you have a single amino acid inserted or deleted, or maybe a number. So you may be able to surmise, due to the sequence, that the amino acid that's been inserted or deleted, that it's likely to have an impact. But I don't know of any good way of looking at these other than examining co-inheritance within the affected family and hoping that you've got more than one member with the variant. Frameshift and nonsense variants are often pathogenic, and can lead to an insertion or deletion interrupting the protein reading frame, which often leads to a null allele. And nonsense variants mostly result in nonsense-mediated decay and lack of protein production. So these variants are often pathogenic, but exceptions can include nonsense variants in the final exon of a gene, And a few examples of this exist, possibly not in the factor 8, factor 9 von Willebrand factor as yet, but in some of the genes that we analyse. For splice site prediction, there's again a number of tools that can be used to help predict possible effect on splice sites. In the UK, commonly, several of the labs use Alamut as our tool of choice for helping to predict pathogenicity, which has just become really expensive. It's gone up from £3,000 a year to £20,000 a year within the space of a couple of months. So they're making lots of money out of it. But it does give us access to a number of tools that can be used together for helping with our pathogenicity predictions. And there are a number of other splice site prediction tools that are also useful if you're not achieving a consensus when using this panel. So again, the aim is to try and use at least 3 different splice prediction tools and hopefully to reach a consensus of potential pathogenicity. We also use splice site prediction for all of our missense variants as well, or the ones that appear to be missense variants, just in case they may actually affect splicing, and we do find a small number where this happens. So this is a family with von Willebrand disease, and having sequenced the gene, we identified a nucleotide substitution at position 55, G to A, which was predicted to lead to a glycine to arginine missense mutation. But when we had a look at the position of the substitution, it's just on the exon 2-3 splice site boundary, and it's actually predicted to lead to a splicing change. So it's not actually acting through being a missense mutation at all. So this is one that's partially penetrant. So in this family, all the individuals with the change are affected, but in other families, not always. So this variant in the homozygous form causes type 3 von Willebrand disease, but it's a partially penetrant type 1 mutation in the heterozygous form. For messenger RNA analysis, we can use this to confirm or exclude the pathogenicity of possible splice site variants if we're not sure about them from our predictions, and can use a fresh blood sample from the patient, extract messenger RNA from the white cells, sequence the region of interest or the entire cDNA, and then look for any differences in the splice product to that of the wild type, and this can help indicate likely pathogenicity. As I mentioned earlier, functional studies may have been helpfully done by somebody else to help determine pathogenicity, or occasionally we can look at those ourselves. We want to make sure that the system being used is relevant for the cell type, and that, for example, for von Willebrand factor, the system used could enable storage in Weibel Palade-like bodies, where we may see that variants like this annoying one of ours actually has no impact at all, but many others will. Co-segregation can be used in the disease in a family to try and help see if there's likely pathogenicity when there's no helpful previous literature or database reports of the variant. So the family analysis may be able to provide information on segregation of the variant and the disorder. And lastly, we may have de novo variants. So we know in haemophilia that these pop up very frequently. So we may have a new variant with no prior family history of the disorder, and where in this instance the mother appears not to have any symptoms. So she could be, in the case of a haemophilia family, a carrier, but she's the first individual with a variant. We can have instances where a parent's a mosaic for a variant and a wild type allele, but it's difficult to detect that in the individual, so it appears as a brand new instance of the variant. We can have the de novo variant just in our patient, and we could also have non-paternity in these families. So there are a number of scenarios to look at. So, to summarise, systematic analysis of several evidence types can provide classification of a sequence variant. For some variants, insufficient evidence is available to help us determine pathogenicity, and they'll be classified as our Class 3 variant of unknown significance, where we'd suggest to the referring clinician that if at all possible, we could get further family members to try and look at co-segregation, which may help to demonstrate possible pathogenicity in that family. And I'll leave it there.